Imagine, imagine for a second that you're trying to navigate a massive, chaotic, completely foreign city.
Good luck with that.
Right. But like the only tool you have is this blurry satellite photo taken from space. You know there are buildings down there, and you can sort of make out the gray streaks of major highways.
But you have absolutely zero idea how the neighborhoods actually function.
Exactly. You don't know where the supply lines are, you can't see the communication hubs, and crucially, you have no way of knowing where the danger zones are hiding.
Which is, I mean, that's terrifying.
It is. And for a very long time in oncology, that blurry photo is exactly how doctors and scientists had to look at glioblastoma, or GBM. It's the most aggressive, most lethal form of primary brain tumor we know of.
Yeah, if you were looking at an MRI, you could clearly see the mass. You knew it was devastating.
Right.
But the intricate street-level organization of the tumor itself, like how the cells were actually interacting with each other in real time, that was essentially invisible. It functioned as a biological black box.
But that black box is finally being pried open. Today, we're taking a deep dive into a groundbreaking February 2026 paper published in Nature Communications.
Such an important paper.
It really is. It's titled A Spatially Resolved Human Glioblastoma Atlas. And our mission for this deep dive is to take you on a high-resolution Google Street View tour of a brain tumor, guided by this new research.
We're going to uncover exactly how this tumor builds its own twisted neighborhoods, how it physically hides from the immune system, and
how it actively brainwashes healthy, supportive cells to act as its own private security force.
Yeah, that part is wild.
Okay, let's unpack this.
So to really understand how revolutionary this street view is, we first have to talk about the monumental leap in technology that made it possible.
The camera upgrade, basically.
Exactly, the camera upgrade. Because historically, analyzing the genetics of a tumor was a profoundly destructive process. If you wanted to know what was inside a biopsy, you essentially had to take that tissue sample and grind it up to sequence the DNA and RNA.
Which, if we go back to our city analogy, is like taking an incredibly complex jigsaw puzzle of the city, throwing all the pieces into a blender, pouring out the resulting dust, and then trying to guess what the picture was.
It's a complete mess.
You know what pieces went into the blender, sure, but you've completely destroyed the architecture. You have no idea how those pieces connected to each other.
Right, you lose all spatial context.
Right.
You might know you have 1 million immune cells and 1 million cancer cells, but you don't know if they were fighting each other or ignoring each other.
Which is a huge problem.
A massive problem.
Mm-hmm.
But this study, throughout the blender, they used a technology called spatial transcriptomics.
Let's define that for a second, because transcriptome is a pretty heavy word.
It is, yeah.
If the DNA in a cell is the master blueprint, like the massive reference library of every single thing the body could possibly build, then the RNA, the transcripts, are the specific photocopied pages that a cell has checked out of the library today to actually build something on the factory floor.
That is a perfect way to look at it. The transcriptome tells us what the cell is actively doing right now.
Right.
And the spatial part means they can read those active blueprints while the cell is still sitting in its exact original location in the tissue.
Wow.
Specifically, this team used 2 advanced platforms side by side to do this: Visium and Xenium. Visium allows researchers to look at gene expression in 55-micron spots across a slice of tissue.
So, staying with the map idea, Visium is giving us the view of individual city blocks. We can see the general zoning laws of a specific neighborhood.
Precisely. It gives you the broad architecture, but then they layered the Xenium platform directly over it, which is where things get really staggering.
Because Xenium goes even deeper.
It offers subcellular resolution. It doesn't just look at the city block, it lets us look through the windows of the individual houses and literally read the text messages the specific residents are sending to each other.
That is wild.
We can see the exact transcripts inside a single cell.
And the sheer scale of the data they pulled from this is hard to wrap your head around. The researchers generated an astounding 115,914 individual spatial transcriptomes.
Over 100,000.
Yeah, and they mapped this across 32 different tissue slices. But what really caught my eye wasn't just the sheer volume of data, it was the type of tissue they used.
Oh, this is a huge point.
They didn't just map perfectly preserved, fresh frozen tissue, did they? They also successfully mapped FFPE tissue.
Yes, formalin-fixed paraffin-embedded tissue.
Which sounds incredibly technical, but if I'm not mistaken, that's just the standard, everyday way hospitals preserve biopsy samples worldwide.
Yep.
They basically fix the tissue in chemicals and embed it in a block of wax so it can sit on a shelf for decades.
It's the ultimate archival method, yes. And proving that this ultra-high-resolution mapping works on standard clinical FFPE samples is a massive win.
Because it's practical.
Exactly. If a new technology only works on fresh, specially frozen tissue that has to be rushed from an operating room to a multi-million-dollar lab on dry ice, well, it's very hard to use in an everyday clinical setting.
Obviously.
But by validating this on the standard wax blocks that every hospital already has sitting in their archives, they've just opened the door for this kind of mapping to eventually become a routine diagnostic tool for anyone, anywhere.
So instead of just a genetic smoothie, doctors could soon pull a standard biopsy, run it through this tech, and know exactly who is living next door to whom, down to the exact street address.
And because of that spatial awareness, the paper notes, they identified 56 distinct cellular states.
56.
It's not just cancer versus immune anymore. It's an entire, highly organized ecosystem.
So if you're an oncologist looking at this new map, your entire battle strategy has to change. Because you wouldn't try to dismantle a hostile city without knowing where the supply lines are. I mean, think about a battle strategy for a second. If you were trying to dismantle a hostile city, wouldn't your first step be figuring out exactly where their supply lines and communication hubs are located?
And that leads us perfectly into the geography of the tumor itself. Because once you have the high-resolution map, the first thing you notice is the massive divide between the dense downtown of the tumor, meaning the core, and the outer edges, the infiltrative periphery.
The suburbs, essentially.
Right. Naturally, the core is where you find the absolute highest density of tumor cells. It is crowded, chaotic, and aggressively expanding.
Sounds awful.
It is. But the defining feature here is the immune system's layout. The immune landscape in the core is entirely alien compared to the periphery.
It's night and day. The paper points out that the core is completely packed with something called blood-derived hypoxic macrophages. Let's break that down. Macrophages are essentially the immune system's garbage trucks, right? They swallow up dead cells and pathogens. But hypoxic means they are oxygen-starved. How is a massive immune response surviving without oxygen?
It comes down to the tumor's chaotic growth. A glioblastoma grows so incredibly fast that it literally outpaces its own blood supply.
Oh, wow.
Yeah, the blood vessels it builds are leaky, dysfunctional, and they just don't reach everywhere. So the center of the tumor becomes this toxic, low-oxygen wasteland.
A hypoxic zone.
Exactly. The tumor then sends out chemical distress signals directly into the bloodstream, actively recruiting specialized macrophages that are perfectly adapted to survive and operate in that exact muck.
Meanwhile, if you look at the map and travel out to the suburbs, the periphery of the tumor, those hypoxic macrophages disappear.
They're gone.
Instead, the area is patrolled almost entirely by resting microglia.
Microglia being the brain's normal, native immune cells. The local police force, basically.
So the core is dominated by these highly specialized imported cells from the bloodstream, while the edges are still mostly relying on the brain's everyday immune system.
But there's another critical discovery in this geographical divide, and it goes back to our earlier conversation about the cameras, specifically how they process the tissue.
Right. The fresh versus frozen tissue comparison. I love this detail because it shows how our own scientific methods have been accidentally blinding us.
It's fascinating. The researchers had the rare opportunity to process some samples completely fresh, literally on the exact same day the tissue was surgically removed from the patient.
Which is super rare.
Very. And when they mapped those fresh samples, they found a huge population of neutrophils.
And neutrophils are the immune system's absolute first responders. They are a type of white blood cell that swarms a site of injury or infection to release toxic chemicals and trap pathogens.
Yes. And in the freshly processed tissue, these neutrophils made up a highly significant 4.6% of the total cells. But when they looked at the exact same type of tumor tissue that had gone through the standard freezing process, the neutrophils only made up 1%.
Because the simple act of freezing the tissue was destroying them. They are so fragile that they burst or degraded before we could even count them.
Exactly.
So for years, all these older sequencing studies were essentially looking at a crime scene where the first responders had already evaporated into thin air. We didn't even know the tumor had this massive rapid response security force constantly moving through it.
It highlights a huge blind spot in our historical understanding of glioblastoma. By capturing them in the fresh samples, this atlas proves that there is an incredibly active, real-time inflammatory response happening that we are systematically underestimating.
So glioblastomas are not uniform blobs. They are highly organized ecosystems. And if the downtown core and the suburban periphery have completely different immune profiles with different oxygen levels, different macrophages, hidden neutrophils, it totally shatters the idea of a one-size-fits-all treatment. I mean, think about it. If you're looking at this new data, how does that change the way you think about delivering a single, uniform cancer drug to the whole brain? How can you deliver a single drug intravenously and expect it to work the same way in a toxic low-oxygen core as it does in the well-oxygenated edges?
You can't. The cellular environment will dictate how the drug is metabolized, or whether it can even physically reach the cells. Which brings us to the actual physical structures of those neighborhoods.
Right.
Knowing the core and periphery are different is just looking at the city zones.
Right.
Now we have to look at the fortresses the tumor is building inside those zones.
And here's where it gets really interesting. This is where the tumor's behavior goes from chaotic to highly calculated. Inside these broader regions, the tumor actively constructs specialized districts designed specifically for its own survival.
The researchers actually managed to map 10 specific anatomical features.
10 of them?
Yeah. They found areas of microvascular proliferation, where the tumor frantically builds new chaotic blood vessels and perinecrotic zones, which are the areas right next to dead tissue.
But two specific micro-neighborhoods stand out as critical therapeutic targets, right? The Immune-Glial Niche, or IGN, and the Immune Fibroblast Niche, or IFN.
Exactly. Let's look at those.
Okay, let's decipher that jargon, starting with the IGN, the Immune-Glial Niche. The map shows this is a dense, highly active hub.
Very dense.
The researchers found it packed with GBM-associated vascular cells, mesenchymal tumor cells, and reactive astrocytes. I want to pause on reactive astrocytes for a second.
Good idea.
Astrocytes are normally the star-shaped cells that hold the brain's physical structure together, right? Like the scaffolding?
That's right. They provide nutrients to neurons and maintain the blood-brain barrier.
Okay.
But when there is trauma or disease, like a massive tumor invading the space, they go reactive. They swell up, change their gene expression, and try to contain the damage.
And the tumor cells in this neighborhood are mesenchymal, meaning they've essentially shapeshifted.
Yes, they become very dangerous.
They've dropped their normal cellular anchors and become highly mobile and aggressive, able to invade deeper into the brain tissue.
Right.
So the IGN is this tense battleground of structural cells panicking and aggressive cancer cells pushing outward.
It's a literal war zone.
But the other neighborhood, the IFN, the Immune Fibroblast Niche, this one is fascinating because of what isn't there.
Precisely. The IFN was mapped using that ultra-precise Xenium data looking inside the houses.
Right.
And they realized that this specific niche lacks glial cells entirely. No astrocytes, no normal brain structural cells.
Not at all.
Instead, it is overwhelmingly packed with fibroblasts and immune cells.
Now, fibroblasts are the cells in our body responsible for building the extracellular matrix. They literally secrete the collagen and proteins that make up the structural framework of our connective tissues.
Exactly.
Because of this, the paper explicitly compares this IFN neighborhood to a fibrotic scar. But I have to push back on that analogy.
Oh.
Yeah, when I think of a scar, I think of healing. I get a cut, fibroblasts rush in, they lay down collagen, the wound closes, and I'm protected. How does the biological mechanism of healing help a lethal brain tumor?
It is a phenomenal question, and it gets to the core of why glioblastoma is so deadly. If we connect this to the bigger picture, the tumor is actually hijacking the body's natural wound healing process and turning it into a weapon.
Wow.
A tumor has frequently been described in pathology as a wound that never heals. The tumor causes tissue damage, which triggers the body to send in fibroblasts to patch the hole.
Makes sense.
But the tumor continuously secretes chemical signals that keep those fibroblasts in overdrive.
So they just keep laying down more and more collagen.
Exactly. They create this incredibly dense, fibrotic scar tissue directly around the tumor cells. And this creates both a physical and a chemical shield. It's a bunker.
A sanctuary site.
A sanctuary site.
Yeah.
Physically, the collagen network is so tight that it prevents large immune cells from penetrating it to attack the cancer.
Like a concrete wall.
Yes. It also compresses the local blood vessels, which means therapies like chemotherapy simply can't flow into the area in high enough concentrations.
That's terrible. Chemically, this dense tissue creates an intensely immunosuppressive environment, secreting signals that actively tell the immune system, to stand down.
That is deeply sinister. It's taking the very mechanism designed to keep us whole and weaponizing it into a bunker.
It is.
So tumors construct specialized micro-neighborhoods, like the IgN and IFN, which mimic wound healing to create physical and chemical shields against the immune system. If a treatment manages to wipe out 99% of the tumor, a tiny reservoir of cancer cells can literally just hide out inside this fibrotic scar, wait out the chemotherapy storm, and then eventually drive the tumor to regrow.
That's exactly how recurrences happen.
Which makes you ask, if a tumor is basically acting like a wound that refuses to heal, should future treatments focus on attacking the scar tissue just as much as the cancer cells themselves?
Yeah.
We have to figure out a way to dissolve the concrete bunkers they are hiding in. That is exactly the therapeutic shift this atlas is pointing us toward. You have to dismantle the fortress. But as the researchers looked even deeper inside these heavily guarded scar-like neighborhoods, they found something even more alarming than the bunkers themselves.
Hard to imagine.
The most important residents keeping these neighborhoods running aren't just cancer cells. They are cells that are fundamentally supposed to be the good guys.
This was the part of the paper that genuinely made me stop and reread the page. We have to introduce the oligodendrocyte.
Yes, we do.
In a normal, healthy brain, an oligodendrocyte is basically the ultimate electrician. Its entire biological purpose is to reach out and wrap our neurons in a fatty substance called myelin.
That myelin acts as crucial insulation. Without it, the electrical signals our neurons use to communicate would leak out or misfire.
Like bare wires.
Exactly. They are absolutely vital for every single normal neurological function you have.
Right. But when the researchers zoomed in on the tumor core, specifically right inside that chaotic immune-glial niche we just talked about, they didn't find normal electricians.
No, they didn't.
They found a bizarre, highly specific mutant subtype of oligodendrocyte. They designated it oligo-232.
It is entirely accurate to think of it as a traitor cell.
A crater cell.
This specific subtype has completely abandoned its post. It is non-myelinating. It has entirely stopped doing its normal job of insulating the brain's wiring.
Unbelievable.
Instead, it has become highly immune-activated. And here is the vital spatial context. This mutant cell is found exclusively in the tumor core. You do not find it out in the healthy periphery.
And thanks to the spatial mapping, we don't just know where it lives, we know exactly who its neighbors are.
We know exactly who it hangs out with.
The map shows this traitor cell physically surrounding itself with those imported immunosuppressive macrophages we talked about and those aggressive, shape-shifting mesenchymal tumor cells.
The bad crowd.
Yeah, it's exactly like an electrician who suddenly stops insulating the city's power grid, quits their job, and instead starts building high-voltage electric fences to protect an organized crime syndicate.
It's actively contributing to the immunosuppressive microenvironment. It is pumping out signals that help the tumor hide from the immune system.
And I imagine the researchers wanted to know just how bad this betrayal was for the patient.
Oh, absolutely. So they didn't just look at their own tissue samples. They cross-referenced this unique traitor cell signature with massive established genomic databases, specifically the TCGA and the CGGA.
And those databases contain the genomic data and clinical outcomes of hundreds and hundreds of glioma patients.
Right.
So let me guess. If this cell is actively guarding the tumor and building electric fences, I have to imagine that patients whose tumors have a high population of these oligo-232 cells do significantly worse.
The correlation was stark and undeniable. Patients who exhibit high levels of this specific signature have significantly poorer overall survival rates.
That is devastating.
The presence of this cell is heavily associated with rapid tumor recurrence and vicious therapy resistance. Even when the researchers statistically adjusted the data for patient age and known aggressive genetic mutations, the mere presence of this traitor cell stood as an independent predictor of a drastically worse outcome.
So glioblastomas reprogram healthy insulating cells into a mutant traitor subtype that actively helps the tumor resist treatment and predicts poor patient survival.
Yes.
I mean, what stands out to you, the listener, the most about the fact that a healthy neurosupportive cell can be brainwashed into becoming a tumor's ultimate bodyguard? The tumor isn't just surviving the body's attacks, it is actively recruiting the brain's own support staff and brainwashing them to fight on the front lines.
It's insidious.
The body doesn't even know it's fighting an enemy because the enemy is wearing the uniform of the local workforce. And the more successful the tumor is at that brainwashing, the faster the patient declines.
It forces a massive paradigm shift.
Yeah.
A tumor like this isn't just a foreign invader sitting in the tissue, it's a systemic corruption of our own biology. It is actively reprogramming the host. But the mechanism of that reprogramming connects to an even larger medical mystery.
Which brings us to the final and perhaps most mind-bending piece of this puzzle. So what does this all mean?
Let's summarize.
We started with the problem of navigating a chaotic city with a blurry satellite photo. By upgrading our cameras to spatial transcriptomics, reading the individual RNA blueprints of cells in their exact locations, we finally have a street-level, high-definition view of a glioblastoma ecosystem.
We really do.
We've seen how the dense, oxygen-starved downtown core imports specialized blood cells while the suburbs rely on the local police. We've mapped the fortified fibrotic bunkers where the tumor hides from chemotherapy, and we've identified the ultimate traitor, the oligo-232 cell guarding the gates.
It's a completely new way of seeing the disease.
It is. Beating this disease clearly isn't just about finding a magic bullet for a single genetic mutation. It's urban warfare. We have to break up the bunkers and deprogram the traitor cells.
And this raises an important question, which is the final thought we should explore.
Let's hear it.
The researchers noted something incredible about these traitorous oligo-232 cells. This exact type of rogue, immune-activated, non-myelinating oligodendrocyte doesn't just exist in brain cancer.
Wait, really?
It perfectly mirrors a cellular profile known as disease-associated oligodendrocytes.
Yeah.
And the place we have thoroughly documented those cells before is in models of neurodegenerative diseases.
Oh, wow.
Specifically, multiple sclerosis.
Multiple sclerosis, an autoimmune disease where the body's immune system mistakenly attacks its own nervous system, stripping away that exact same myelin insulation.
Yes.
The fact that a lethal brain cancer and a degenerative autoimmune disease are somehow utilizing the exact same corrupted cell type is staggering.
It is the exact same biological playbook being used for two wildly different outcomes. In MS, the corrupted cell contributes to the destruction of the brain's wiring. In glioblastoma, the corrupted cell protects the lethal mass. But if the mechanism of corruption is the same, could the cure for one hold the key to the other? What if we don't need to invent a brand new drug from scratch to stop this traitor cell?
That would be amazing.
What if the next great breakthrough in cancer treatment isn't going to be found in an oncology lab at all? It might currently be sitting in a completely different aisle of the pharmacy among the treatments for multiple sclerosis, just waiting to be repurposed.
That is exactly the kind of perspective shift this map provides. As we finally get a clear molecular view of this chaotic, deadly city, it turns out the key to dismantling its defenses might just be understanding the broken roads we've already mapped somewhere else.